So, very good morning, dear students. So, today we'll be starting with the second chapter. See, in the previous class, we had completed reproduction in organisms, isn't it? And we had discussed asexual reproduction and sexual reproduction. So, in today's class, we are going to discuss about sexual reproduction in flowering plants. So what we are going to study under the sexual reproduction in flowering plants is we'll be studying the structure of the Microsporangium, Anther wall, the different layers of Anther wall. So we are also going to study about the formation of pollen grain. So which involves two steps. One is microsporogenesis, another is microgametogenesis. They don't use these terms, but for you to understand it and remember it, it would be easy if you remember them by these terms. Microsporogenesis is formation of microspore from microspore mother cell. So the microspore mother cell undergoes meiosis to form microspores, that is the pollen grain. Within the pollen grain, they undergo cell division. So that we call it as that cell division is mitotic, and we call that as microgametogenesis. After this microgametogenesis, there is going to be the formation of two sperm cells and which will be present within the vegetative cell. So three-celled pollen grain, two-celled pollen grain, you can notice after microgametogenesis. So all these are things that we'll be discussing in today's session. So it is an important aspect to understand there is a sexual reproduction in flowering plants also. So in the first slide that you notice here, so you can notice this is, can you identify which flower it is? You can switch on your mic
Hibiscus.
and ask.
Hibiscus.
Hibiscus. Yeah, that's what we call it as hibiscus rosa sinensis or China rose. So you can notice the monoadelphous stamens. They are fused to form a single bundle, isn't it? So they have a kidney-shaped anther, and on top of it, you can notice this yellow powder. That is the pollen grains are there. And you also notice here pentaphyt stigma. If you dissect this stalk, this corolla tube, you can see this pentaphyt stigma is having a style, a slender style, which is passing through the staminal tube and it reaches the ovary. So they have here pentaphyt stigma. So the pollen grains of another hibiscus flower, when it is brought by an insect and shed on this pentaphyt stigma, they germinate and they produce the pollen tube, which enters into the style, ovary, and ovule. That we'll discuss later on. Okay. So now just see in the slide the pollen grains on the anther. So the pollen grains are liberated here. So they are just waiting for the insects to uh, visit the flower and get the pollen grains dusted on their insects abdomen or on their thorax. They get dusted and the pollen grains are carried by the insects. So this is what you notice in this hibiscus. So our topic for today's discussion is sexual reproduction in flowering plants. See, all the flowering plants in angiosperm, whether it is monocot or dicots, they exhibit sexual reproduction, and flowers are the sites of sexual reproduction. So where does this sexual reproduction takes place in angiosperms, whether it is monocot or dicot? So the region where sexual reproduction takes place is flowers. So ಅದಕ್ಕೆ ಒಂದು flower definition ಇದೆ. Flower is a modified vegetative shoot meant for the purpose of sexual reproduction. Flower is a modified vegetative shoot meant for the purpose of sexual reproduction. So that definition you should be knowing about. So where is the sexual reproduction going to take place? It is going to take place within the flower. So you know that in the first year you have studied flower has four whorls. The four whorls are the outermost whorl is calyx. Calyx is collection of sepals. The second whorl is corolla. Corolla is collection of petals. The third whorl is andrecium. Andrecium is a collection of stamens, and you know that stamen is made up of filament and anther. Okay, and the gynoecium is collection of carpels. See, andrecium is a male reproductive organ, gynoecium is a female reproductive organ. So they are the essential whorls. Calyx and corolla are accessory whorls or non-essential whorls. They are not involved in reproduction, so we call them as non-essential whorls, whereas calyx and corolla are accessory whorls or non-essential whorls, but andrecium and gynoecium, they are essential whorls. They take part in sexual reproduction. So in the left slide, you can see an inflorescence. Inflorescence is collection of flowers on this inflorescence axis. So you notice this umbel inflorescence, the example is. So it is a collection of flowers on the inflorescence axis. Inflorescence axis, we call it as peduncle. So the pre-fertilization events in case of flowers, the structures and events that are taking place before fertilization is several hormonal and structural changes. They result in differentiation and development of the floral primordium. So you can notice this below slide where there is a carpel primordium, stamen primordium, sepal primordium, petal primordium. So they are all at the initial stages. So several hormonal and structural changes results in the differentiation and development of the floral primordium. So the floral primordium differentiates to form sepal, petal, andrecium, and gynoecium. That is brought about by changes in the hormone and structure. Okay, so and they develop to form the floral bud. Moggagate. So the inflorescence bear the floral buds and then the flowers. So inflorescence is a collection of flowers. Alva. Initially they'll have floral buds and then they bloom. Arulute, hooagadike, and then they bloom to form the flowers. So the pre-fertilization discussion, you should also know the structure of a flower. So a typical flower has two parts, the andrecium and gynoecium. Essential whorls we call it as. Andrecium is the male reproductive organ and it is made up of stamens. Gynoecium or pistil, it is the female reproductive organ of a flower. So if you enlarge this andrecium, one stamen and observe it, so they have a filament and they have an anther. Anther ಅಲ್ಲಿ ಎರಡು lobe ಇರುತ್ತೆ. They have two lobes. So if they have two lobes, we call them as dithecous anthers. The point at which filament connects with the anther, we call it as connective. The point at which filament connects with the anther, we call it as connective. So ಈ ಎರಡು ಲೋಬ್ ಇರುತ್ತೆ, two lobes in the anther, and if you see the inner contents of this lobe, they would be having the pollen grain. So the anther lobes, we call them as microsporangium. So anthers, we call them as microsporangium. So remember about that. So within this microsporangium, you find the microspores or pollen grains. That is in case of andrecium. So gynoecium, you just if you enlarge this gynoecium, so they have a ovary, they have style and stigma. Stigma is the region where the pollen grains fall and they germinate. The pollen grains falls and germinates to produce pollen tube. Style is a long stalk-like region called as style. Within the ovary, they have ovules. So ovules are called as megasporangia. So ovules are called as megasporangia, and within these ovules they have the embryo sac. Embryo sac has the egg apparatus in the micropylar region and antipodal cells in the chalazal region. So we'll study about that later. So this entire chapter it deals with formation of pollen grain, formation of egg cell. So there we are going to study in formation of pollen grain, microsporogenesis, microgametogenesis. In the formation of embryo sac, we are going to study two concepts. that is Megasporogenesis and Megagametogenesis. So we'll discuss that later. Now just understand a typical structure of gynecium or pistil. Gynecium is a collection of carpels, you know that. So the gynecium has a stigma and a long stalk called as style. So style leads to ovary. Ovary has ovules. Ovules are also called as megasporangia. So the androecium, it is the male reproductive part of the flower. It consists of a whorl of stamens. The stamen number and length, they vary from species to species. So you can see this uh, stamen which has this uh, dithecous anthers with a filament. So when you dissect that, you find that the pollen grains are found within this microsporangium. So the pollen grains are called microspore. Okay, the anther lobes are called as microsporangium. The parts of a stamen are filament and anther. The point at which the filament connects to the anther, we call it as collective. Okay, the point at which the filament connects to the anther, this point we call it as connective. See, the filament is connecting to the anther at this point. We call that as filament. So in the structure of a flower, so what you can notice is in the structure of a flower, the filament we will discuss about. Filament is a long and slender stalk. So filament is going to be a long and slender stalk. Its proximal end, that is its base, is attached to the thalamus or the petal of the flower. See, if it's proximal end of the filament, if it is attached to the petal of the flower, we use a term called as epipetalous. So if the proximal end of the filament it is attached to the petal of a flower, we call it as epipetalous. If it is attached to the thalamus, there is no name. So just it is attached to the thalamus. If the proximal end of the filament is attached to the petal of a flower, we call that condition as epipetalous. So remember about that. So filament is long and slender. You can see it is a very long and slender stalk. It connects the anther at the connective. Okay, so the proximal end of this filament, it is usually attached to the thalamus, or in some cases they are attached to the petals of a flower. Then we call that condition as epipetalous condition. So the anther, the terminal and typically bilobed anther, you can see each lobe has two cavities or theca. So that is the reason anthers are called as dithecous, because they are two lobes. So we call it as bilobed or dithecous. Each lobe has uh, the dithecous part of it. Often you can also notice a longitudinal groove runs lengthwise separating the theca. So theca is the cavity. So you can notice that. So the bilobed, each lobe has two thecas and they are here separated by the longitudinal groove inside. So two thecas per lobe. So totally four you can notice. So the dithecous condition you can notice here. When you take a section of an anther, I always call this as butterfly diagram. So when you take a transverse section of an anther and see this diagram, they would be uh, giving it for labeling during your NEET examination. So diagram oriented question they would be asking you. Okay, so section of dithecous anther when you take, you find it to be tetragonal structure which consists of four microsporangia, tetrasporangiate, you can call it as. So the section of anther shows four microsporangia located at the corners. So there are two microsporangia in each lobe. So there are two microsporangia in each lobe. So the microsporangia develop to form the pollen sac. They extend longitudinally all through the length of an anther, and they are packed with pollen grains. So the microsporangia develop to form the pollen sacs, which extend longitudinally. See in the second diagram, you can see this Microsporangia extends longitudinally to form an pollen sac, and this anther is packed with pollen grains. So you can notice this pollen sac formed by the extension of this microsporangia longitudinally. So the structure of microsporangium. So when I'm talking about microsporangium, it is the structure of anther we are talking about. If you take a thin section and observe it under microscope, see in the left side, you can see this is a permanent slide of structure of microsporangium or anther. So what you notice is it is made up of four layers. The microsporangium or the anther wall is made up of four layers. The first layer we call it as epidermis. Second layer is endothelium. It has fibrillar thickenings. So you can see here the diagram that is given taken from your textbook. So the first layer is the epidermis. Second layer is the endothelium. Third layer is the middle layers. Then the innermost layer you have the tapetum, and within this tapetum you find the sporogenous tissue. So the outer three layers give protection. The outermost three layers, that is epidermis, endothelium, and middle layers, they give protection, and whereas the and also help in dehiscence of anther to release the pollen grain. So epidermis, endothelium, and middle layers, they provide protection to the uh, anther, and they also help in the dehiscence of the anther to release the pollen grain. So you have to look into the labeling. See, the connective is parenchymatous tissue you have. This is the ground plan of an anther wall or microsporangium. So they have this butterfly like diagram I have always told you. So you can see four lobes. We call it as four microsporangium or tetrasporangiate condition. You can see at the center they have a connective tissue. See, the outermost layer of this microsporangium or anther wall is epidermis. Second layer is endothelium, which has fibrillar thickenings and bring about dehiscence of anther. So the inner layer is the middle layers. It might be one in number or two in number, or in some of the anther walls, middle layer is absent. Accordingly, we call it as if they have one middle layer, unitegmic. If they have two middle layers, bitegmic. If middle layers are absent, ategmic. So these are terminologies that we have. Then the innermost layer is the nutritive tissue, that is the tapetum. It provides nourishment for the developing sporogenous tissue. So a small diagram of microsporangia. Is anther wall. So have this middle layers. So the first three layers, epidermis, endothelium, and middle layers, they are protective in function, and they also help in the dehiscence of the anther and release of the pollen grain. So the first three layers, they do that. So the nutritive tissue is the tapetum, which provides nutrition for the developing sporogenous tissue. So this diagram labeling ನೀವು ನೋಡ್ಕೋಬೇಕಾಗತ್ತೆ. ಈ ಡಯಾಗ್ರಾಮ್ ವಿತ್ ಕರೆಕ್ಟ್ ಲೇಬಲಿಂಗ್ ಡಯಾಗ್ರಾಮ್ ಓರಿಯೆಂಟೆಡ್ ಕ್ವಶನ್ಗೆ ಕೊಡ್ತಾರೆ. ಸೊ ಇದು ಮುಖ್ಯ. ಇದನ್ನ ನೋಡ್ಕೊಳ್ಳಿ ನೀವು. ಡಯಾಗ್ರಾಮ್ನ ಲೇಬಲಿಂಗ್ನ ಸ್ವಲ್ಪ ತರವಾಗಬೇಕು. So now when we study the structure of microsporangium, the tapetum is the innermost layer and they nourish the developing pollen grains. The tapetum is the innermost layer. which nourishes the developing pollen grains. And if you look into the cells of the tapetum, they contain dense cytoplasm and generally have more than one nucleus. So the tapetum is going to nourish the developing pollen grains. That is the function of tapetum. So if you look into the cells of the tapetum, they contain dense cytoplasm and they have more than one nucleus. That is a feature of tapetum. Okay, it is almost like yolk of the cell. ಈ ಎಗ್ಗಲ್ಲಿ ಹೇಗೆ ಯೋಕ್ ಇರುತ್ತೋ ಡೆವಲಪಿಂಗ್ ಎಂಬ್ರಿಯೋಗೆ ನ್ಯೂಟ್ರಿಷನ್ ಕೊಡುತ್ತೋ ಅದೇ ರೀತಿ ಟೆಪಿಟಮ್ ಬಂದು ಈ ಸ್ಪೊರೋಜಿನಸ್ ಟಿಶ್ಯೂ ಡೆವಲಪ್ ಆಗೋದಕ್ಕೆ ಆಹಾರ ನೀಡುತ್ತೆ. ಸೊ ದಟ್ ಇಸ್ ವಾಟ್ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ಪ್ರೊವೈಡಿಂಗ್ ನ್ಯೂಟ್ರಿಷನ್ ಫಾರ್ ದಿ ಡೆವಲಪಿಂಗ್ ಸ್ಪೊರೋಜಿನಸ್ ಟಿಶ್ಯೂ. ಸೊ ಈ ಟೆಪಿಟಲ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ಗಿಂತ ಅಧಿಕವಾಗಿ ಇರುತ್ತೆ. ಸೊ ಇಟ್ ಇಸ್ ಹ್ಯಾವಿಂಗ್ ಮೋರ್ ದ್ಯಾನ್ ಒನ್ ನ್ಯೂಕ್ಲಿಯಸ್. ಇನ್ ಯಂಗ್ ಆಂಥರ್ each microsporangium has sporogenous tissue at center. In young anther, each microsporangium has sporogenous tissue at center. It consists of compactly arranged homogeneous diploid cells. So ಈ sporogenous tissues ಇರುತ್ತಲ್ಲ, ಇನಿಷಿಯಲಿ ಇದು ಬಂದು ಡಿಪ್ಲಾಯ್ಡ್ ಸೆಲ್ಸ್. ಓಕೆ, ಸೊ ಹೋಮೋಜಿನಸ್ ಅಂದ್ರೆ ಒಂದೇ ರೀತಿಯಾದಂಥ ಸೆಲ್ಸ್ಗಳಿರತ್ತೆ ಅಲ್ಲಿ. ಇಟ್ ಕನ್ಸಿಸ್ಟ್ ಆಫ್ ಕಾಂಪ್ಯಾಕ್ಟ್ಲಿ ಅರೇಂಜ್ಡ್ ಹೋಮೋಜಿನಸ್ ಡಿಪ್ಲಾಯ್ಡ್ ಸೆಲ್ಸ್. ಇದನ್ನ ನಾವು ಏನಂತೀವಿ ಅಂದ್ರೆ ಸ್ಪೋರೋಜಿನಸ್ ಸೆಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಆ ಇಡೀ ಟಿಶ್ಯೂನ ಸ್ಪೋರೋಜಿನಸ್ ಟಿಶ್ಯೂ ಅಂತೀವಿ. ಸೊ ಇನಿಷಿಯಲಿ ಇದು ಡಿಪ್ಲಾಯ್ಡ್ ಸೆಲ್ಸ್ ಆಗಿರತ್ತೆ. ಸೊ ಆಸ್ ದಿ ಆಂಥರ್ ಡೆವಲಪ್, as the anther develops, each sporogenous cell, microspore or pollen mother cell undergoes meiotic divisions to form microspore tetrads. As the anthers are going to develop, you notice that some of the sporogenous cells they become larger. We call them as microspore mother cell, or you can call them as pollen mother cell. And this pollen mother cell, see in one of these cells, ಈ ಡಯಾಗ್ರಾಮ್ ಅಲ್ಲಿ ನೀವು ನೋಡ್ಬೋದು, ಕೆಲವು ಸೆಲ್ಸು ದೊಡ್ಡದಾಗತ್ತೆ, ನ್ಯೂಕ್ಲಿಯಸ್ ದೊಡ್ಡದಾಗತ್ತೆ. ಅಂಥವನ್ನ ನಾವು ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಅಥವಾ ಪೋಲನ್ ಮದರ್ ಸೆಲ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ದಿಸ್ ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಡಿಪ್ಲಾಯ್ಡ್ ಆಗಿರತ್ತೆ. ಈ ಚಾರ್ಟ್ ಅಲ್ಲಿ ನೋಡಿ, ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ವಿಚ್ ಇಸ್ ಡಿಪ್ಲಾಯ್ಡ್, ದೇ ಅಂಡರ್ಗೋ ಮಿಯಾಸಿಸ್ ಟು ಫಾರ್ಮ್. ಫಸ್ಟ್ ಮಿಯಾಸಿಸ್ ಆದಾಗ ಡಯಾಡ್ ಆಗತ್ತೆ, ಸೆಕೆಂಡ್ ಮಿಯಾಸಿಸ್ ಆದಾಗ ಟೆಟ್ರಾಡ್ ಆಫ್ ಪೋಲನ್ಸ್. ಸರ್ ಎಲ್ಲಿ ಸರ್ ಮೂರೇ ಸೆಲ್ ಇದೆಯಲ್ಲ ಸರ್, ನಾಲ್ಕನೇದು ಎಲ್ಲೋ ಅಂದ್ರೆ ಹಿಂಭಾಗ ಇರುತ್ತೆ ಅದು. ಸೊ ಟೆಟ್ರಾಹೆಡ್ರಲ್ ಅರೇಂಜ್ಮೆಂಟ್ ಆಫ್ pollen grain. Okay, so 4 pollen grains are formed and they are all haploid. ಈಗ ಅವ 4 pollen grain ಟೆಟ್ರಾಡ್ ಆಗಿತ್ತಲ್ಲ, ಅದು ಸಪರೇಟ್ ಆಗುತ್ತೆ. and these pollen grains are going to separate to form haploid pollen grains. ಸೊ ಅದನ್ನೇ ಈ ಸೆಂಟೆನ್ಸ್ ಅಲ್ಲಿ ಹೇಳಿರ್ತೀವಿ, ಆಸ್ ಎ ಆಂಥರ್ ಡೆವಲಪ್ಸ್ ಈಚ್ ಸ್ಪೋರೋಜಿನಸ್ ಸೆಲ್, ಈ ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಇದೆಯಲ್ಲ, ಅಥವಾ ಮೈಕ್ರೋಸ್ಪೋರ್ ಪೋಲನ್ ಮದರ್ ಸೆಲ್ ಅಂತೀವಿ ಅಥವಾ ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್, ದೇ ಅಂಡರ್ಗೋ ಮಿಯೋಟಿಕ್ ಡಿವಿಷನ್ಸ್ ಟು ಫಾರ್ಮ್ microspore tetrads. So the microspores are arranged in a cluster of 4 cells. So the formation of microspores from a pollen mother cell through meiosis. So the formation of microspores from a pollen mother cell through meiosis, we call it as microsporogenesis. So microsporogenesis andre ಏನು? Formation of pollen grains from a pollen mother cell or microspore mother cell. We call that process as microsporogenesis. ಈ microsporogenesis ಅಲ್ಲಿ meiosis involve ಆಗತ್ತೆ. ಈ 4 pollen grains ಆಗಿದೆ ಈಗ, ದಟ್ ಇಸ್ ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್. ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಇಂದ 4 pollen grain ಮಿಯೋಟಿಕ್ ಸೆಲ್ ಡಿವಿಷನ್ ಮೂಲಕ ಆಗಿರೋದನ್ನ ನಾವು ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್. ಜೆನೆಸಿಸ್ ಅಂದ್ರೆ ಫಾರ್ಮೇಶನ್. ಓಕೆ, ಸೊ ಆಸ್ ದಿ ಆಂಥರ್ಸ್ ಮೆಚೂರ್ ಅಂಡ್ ಡಿಹೈಡ್ರೇಟ್, ಈ ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಟೆಟ್ರಾಡ್ ಅಲ್ಲಿ ಇರುತ್ತಲ್ಲ, ಸಪರೇಟ್ ಆಗುತ್ತೆ. ದಿ ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಡಿಸೋಸಿಯೇಟ್ from each other and they develop into pollen grains. Each microsporangium they contain thousands of pollen grains. ಈ pollen grains ಡಿಹೈಸನ್ಸ್ ಆಗೋದ್ರ ಮೂಲಕ ರಿಲೀಸ್ ಆಗುತ್ತೆ. So each microsporangium they contain thousands of pollen grains. So they are released with the dehiscence of anther. Okay. ಸೊ ಈಗ ಆಂಡ್ರೀಷಿಯಂ ಪೋಲನ್ ಗ್ರೈನ್ ಆಗೋದಕ್ಕೆ ಮುಂಚೆ ಇನ್ನೊಂದು ಸ್ಟೆಪ್ ಹೇಳಬೇಕಾಗಿತ್ತು. ಐ ಗೋ ಟು ದಿ ಪ್ರೀವಿಯಸ್ ಸ್ಲೈಡ್. ಈ ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್, ದಿ ಮೈಕ್ರೋಸ್ಪೋರ್ ಆಗುತ್ತಲ್ಲ ಪೋಲನ್ ಗ್ರೈನ್, ದೇ ಹ್ಯಾವ್ ಎನ್ ಔಟರ್ ಮೋಸ್ಟ್ ಲೇಯರ್ ಕಾಲ್ಡ್ ಆಸ್ ಎಕ್ಸೈನ್, ಇನ್ನರ್ ಮೋಸ್ಟ್ ಲೇಯರ್ ಕಾಲ್ಡ್ ಆಸ್ ಇಂಟೈನ್. ಈಗ ಒಂದೇ ಒಂದು ಸೆಲ್ ಆಗಿರುತ್ತೆ ಇದು. ಇನಿಷಿಯಲಿ ಮೈಕ್ರೋಸ್ಪೋರಸ್ ಸಿಂಗಲ್ ಸೆಲ್. Okay. Now this single celled microspore, they undergo cell division, unequal cell division to form a large vegetative cell and a small generative cell. So this microspore or pollen grain, they undergo ಇದನ್ನ ನಾವು ಮೈಕ್ರೋಗ್ಯಾಮಿಟೋಜೆನೆಸಿಸ್ ಅಂತೀವಿ. ಈ ಪ್ರೋಸೆಸ್ನ ನಿಮ್ಮ ಬುಕ್ಕಲ್ಲಿ ಹೇಳಿಲ್ಲ, ಬಟ್ ಈ ಮೈಕ್ರೋಸ್ಪೋರ್ ಅಲ್ಲೇ ಸೆಲ್ ಡಿವಿಷನ್ ಒಳಗೆ ಆಗತ್ತಲ್ಲ, ಅದನ್ನ ಮೈಕ್ರೋಗ್ಯಾಮಿಟೋಜೆನೆಸಿಸ್ ಅಂತೀವಿ. So initially pollen grain will be single celled. So this pollen grain has an outermost exine, innermost intine, they're all there. This pollen grains undergoes mitosis to form unequal mitosis to form a large vegetative cell and a small generative cell. ಈಗ ಪೋಲನ್ ಗ್ರೈನ್ ಇದೇ ಸ್ಟೇಜ್ ಅಲ್ಲಿ ರಿಲೀಸ್ ಆದ್ರೆ ಇದನ್ನ ನಾವು ಟೂ ಸೆಲ್ಡ್ ಪೋಲನ್ ಗ್ರೈನ್ ಅಂತೀವಿ. ಕೆಲವು ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಈ ಜನರೇಟಿವ್ ಸೆಲ್ ಇದೆಯಲ್ಲ, ಇದು ಇನ್ನೊಂದು ಮೈಟೋಟಿಕ್ ಸೆಲ್ ಡಿವಿಷನ್ ಆಗುತ್ತೆ. ಆ ಜನರೇಟಿವ್ ಸೆಲ್ ಅವಾಗ ಏನಾಗುತ್ತೆ, ಎರಡು ಸ್ಪರ್ಮ್ ಸೆಲ್ಸ್ ಆಗುತ್ತೆ. ಸಿ ಇನ್ ಕೇಸ್ ಆಫ್ ಮೋಸ್ಟ್ ಆಫ್ ದೀಸ್ microspores they undergo unequal cell division and they form a large vegetative cell and a small generative cell. So this, if the pollen grain is liberated at this stage, we call it as two-celled pollen grain. But in some of the plants, the generative cell undergoes one more cell division. So that results in the formation of two sperm cells. So we call this as three-celled pollen grain. Three-celled pollen grain has two sperm cells within the vegetative cell. So that is called as three-celled pollen grain. So this process of a microspore maturing into a pollen grain, we call that as microgametogenesis. So initially they would be single cell, later they form three-celled or two-celled pollen grain. They are liberated. ಸೊ ಈಗ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಪೋಲನ್ ಗ್ರೈನ್ ನೋಡಿದ್ರೆ, ಸೊ ಈ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಲ್ಲಿ ಮೇಲ್ಗಡೆ ಸ್ಲೈಡ್ಸ್ ಇದೆ. ದೇ ಆರ್ ದಿ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ಪೋಲನ್ ಗ್ರೈನ್. ಆಚೆ ನೋಡಿ ಏನೇನ್ ಡಿಸೈನ್ಸ್ ಇರುತ್ತೆ, ಪ್ಯಾಟರ್ನ್ಸ್ ಇರುತ್ತೆ. ಸೊ ನೀವು ಇದನ್ನ ನೋಡ್ಬೋದು. ಈ ಕೆಲವು ಸರ್ಕ್ಯುಲರ್ ಆಗಿ ಸ್ಪೆರಿಕಲ್ ಆಗಿರುತ್ತೆ ಪೋಲನ್ ಗ್ರೈನು, ಕೆಲವು ಬಂದು ಈ ಸ್ಪಿಂಡಲ್ ಶೇಪ್ಡ್, ಈ ರಗ್ಬಿ ಫುಟ್ಬಾಲ್ ಆ ಥರ ಇರುತ್ತೆ. sulcus type of pollen grain, spherical pollen grain. So there are different types of pollen grains that you have to remember. Generally, a pollen grain has an outermost layer called as exine and an innermost layer called as intine. Okay, the exine has a chemical deposited on them called as sporopollenin. So this makes the pollen grain very sturdy. Sporopollenin is deposited on the exine. And you can notice within the exine, there are certain regions which we call it as the apertures or pores. So the innermost layer is the intine. ಮತ್ತೆ ಒಳಗೆ ನೋಡಿದ್ರೆ pollen grain ಅಲ್ಲಿ it is a two-celled pollen grain with a vegetative cell having a vegetative nucleus and a generative cell having a generative nucleus. Okay, so pollen grains are generally spherical. ಅವುಗಳ ಸೈಜ್ ಏನಿರುತ್ತೆ ಅಂದ್ರೆ 25ರಿಂದ 50 ಇರುತ್ತೆ ಸೈಜ್. So they are 25 to 50 millimeter in diameter. The cytoplasm is surrounded by plasma membrane. A pollen grain has two-layered wall, outer exine, which is made up of sporopollenin, and inner intine. So they have two walls, an outer exine, which is chemically made up of sporopollenin, and they are sturdy. Pollen grains are sturdy because of this chemical. And inner layer, we call it as intine. ಈಗ ಎಕ್ಸೈನ್ ಸ್ಟಡಿ ಮಾಡಿದಾಗ ಪೋಲನ್ ಗ್ರೈನ್ ಇಸ್ ನಥಿಂಗ್ ಬಟ್ ದಿ ಮೇಲ್ ಗ್ಯಾಮೆಟೋಫೈಟ್. The pollen grain is male gametophyte. It might be two-celled male gametophyte or three-celled male gametophyte. So the exine is the hard outer layer of the pollen grain, and it is made up of sporopollenin. Sporopollenin is a chemical. ಈಗ ನಿಮ್ ನೀಟ್ ಎಕ್ಸಾಮ್ಸ್ ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ ಎಕ್ಸೈನ್ ಇಸ್ ಹಾರ್ಡ್ ಅಂಡ್ ಸ್ಟರ್ಡಿ ಬಿಕಾಸ್ ಆಫ್ ಕೈಟೇನ್, ಸ್ಪೋರೋಪೊಲನಿನ್, ಸೆಲ್ಯುಲೋಸ್, ಪೆಕ್ಟಿನ್ ಅಂತ ಕೊಡ್ತಾರೆ. ಸ್ಪೋರೋಪೊಲನಿನ್ ಇಸ್ ದಿ ಆನ್ಸರ್. ಸೊ ಸ್ಪೋರೋಪೊಲನಿನ್ ಇಸ್ highly resistant to organic material. So sporopollenin can withstand high temperatures and strong acids and alkali. Enzymes cannot degrade sporopollenin. That is the reason fossil pollen grains are still preserved because of the exine. So exine is the outermost layer, hard outermost layer of the pollen grain. It is chemically made up of sporopollenin. So sporopollenin is highly resistant organic material. So this makes the pollen grain withstand high temperature, strong acids, and alkali. ಯಾವುದೇ ಎಂಜೈಮು ಸ್ಪೋರೋಪೊಲಿನಿನ್ನ ಡಿಗ್ರೇಡ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. Enzymes cannot destroy the sporopollenin. So exine has germ as apertures or opening called as germ pores where sporopollenin is absent. So exine is having this germ pores where sporopollenin is absent. It is through this germ pore the pollen tube comes out. Okay, apertures are opening. So the pollen grains are preserved as fossils for a long duration due to the presence of sporopollenin, and exine exhibits various patterns and designs. So it can have a reticulate pattern, it can have a colporate, it can have spinescent type of pattern. So a lot of patterns are there on the exine. That itself is an interesting study, but it is done at degree level. Now you don't require this. So what we have to remember is pollen grain is also called as male gametophyte. So this pollen grain has an hard outermost layer called as exine, which is made up of sporopollenin. So this sporopollenin is highly resistant to organic material. So it can withstand high temperature and strong acids and alkali. So the enzymes cannot destroy this sporopollenin. So exine has numerous apertures or opening called as germ pores where there is no deposition of sporopollenin. ಈ diagram ಅಲ್ಲಿ ನೋಡಿದ್ರೆ germ pores ಹತ್ರ sporopollenin ಇರಲ್ಲ, ಇದ್ರೂ ಕಡಿಮೆ ಇರತ್ತೆ. ಸೊ ದಿಸ್ germ pores ಮೂಲಕ pollen tube ಆಗತ್ತೆ. So pollen grains are preserved as fossils due to the presence of sporopollenin. So pollen grains ಇವೆಲ್ಲ ಫಾಸಿಲ್ ಆಗಿ ಯಾಕೆ ಪ್ರಿಸರ್ವ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ ಸ್ಪೋರೋಪೊಲೆನಿನ್ ಇರೋದ್ರಿಂದ. Exine exhibits patterns and design. Intine ಅನ್ನೋದು inner wall. Intine is the inner wall. It is a thin and continuous layer, and intine is made up of cellulose and pectin. Exine is made up of sporopollenin. Intine is made up of cellulose and pectin. So a mature pollen grain contains two cells or three cells. Majority of the pollen grains they have two cells. That is a vegetative cell. It is bigger, has abundant food reserve, and a large irregularly shaped nucleus. ಈಗ ಈ ವೆಜಿಟೇಟಿವ್ ಸೆಲ್ಲು ದೊಡ್ಡದಾಗಿರುತ್ತೆ, ಫುಡ್ ಮೆಟೀರಿಯಲ್ ಇರುತ್ತೆ, ಎಕ್ಸಸ್ ಫುಡ್ ಮೆಟೀರಿಯಲ್ ಇರುತ್ತೆ, ಮತ್ತೆ ಇರ್ರೆಗ್ಯುಲರ್ ಶೇಪ್ಡ್ ದೊಡ್ಡದಾದಂಥ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ. ಸೊ ದೇ ಹ್ಯಾವ್ ಅ ಲಾರ್ಜ್ irregularly shaped nucleus. So a matured pollen grain contains two cells, a large vegetative cell which is bigger, has abundant food reserve, and a large irregular shaped nucleus is noticed in vegetative cell. Whereas the generative cell is smaller and it floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and nucleus. So ಇಲ್ಲಿ separate ಆಗಿಲ್ಲ ಇನ್ನು generative cell. So generative cell will usually be found floating in the ವೆಜಿಟೇಟಿವ್ ಸೆಲ್. ಅದಕ್ಕೆ ಈ ಕಾನ್ಸೆಪ್ಟ್ನ ಸೆಲ್ ವಿತಿನ್ ಅ ಸೆಲ್ ಅಂತ ಕರಿತೀವಿ. ಸೆಲ್ ಒಳಗೆ ಸೆಲ್. ಜನರೇಟಿವ್ ಸೆಲ್ ಬಂದು ವೆಜಿಟೇಟಿವ್ ಸೈಟೋಪ್ಲಾಸಮ್ ಅಲ್ಲಿ ತೇಲ್ತಾ ಇರುತ್ತೆ. ಸೊ ದಿ generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped. ಈ ಜನರೇಟಿವ್ ಸೆಲ್ ಬಂದು ಸ್ಪಿಂಡಲ್ ಶೇಪ್ ಆಗಿರುತ್ತೆ. ಜೊತೆಗೆ ಡೆನ್ಸ್ ಸೈಟೋಪ್ಲಾಸಮ್ ಮತ್ತೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ. ಓಕೆ, ಸೊ ಈ ಜನರೇಟಿವ್ ಸೆಲ್ಲು ವೆಜಿಟೇಟಿವ್ ಸೆಲ್ ಒಳಗೆ ತೇಲಾಡೋದ್ರಿಂದ ಈ ಪೋಲನ್ ಗ್ರೈನ್ ನ ಸೆಲ್ ವಿತಿನ್ ಅ ಸೆಲ್ ಅಂತಾನು ಕರಿಬಹುದು. ಸೆಲ್ ವಿತಿನ್ ಅ ಸೆಲ್, ಸೆಲ್ ಒಳಗೆ ಇನ್ನೊಂದು ಸೆಲ್. So over 60% of angiosperms, they shed their pollen grains at two cell stage. So 60% of angiosperms, they are going to shed their pollen grains at two cell stage. In others, generative cell divides mitotically to form two male gametes. Thus, these pollen grains are liberated at three cell stage. ಇನ್ನು 40% ಗಿಡಗಳಿದೆ ನೋಡಿ, ಅದರಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಈ ಜನರೇಟಿವ್ ಸೆಲ್ ಇನ್ನೊಂದು ಮೈಟಾಟಿಕ್ ಡಿವಿಷನ್ ಆಗುತ್ತೆ. ಎರಡು ಸ್ಪರ್ಮ್ ಸೆಲ್ಸ್ ಆಗುತ್ತೆ. ಇವು ಎರಡು ಸ್ಪರ್ಮ್ ಸೆಲ್ಸ್ ತೇಲಾಡ್ತಿರತ್ತೆ ವೆಜಿಟೇಟಿವ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ. ಸೆಲ್ಸ್ ವಿತಿನ್ ಅ ಸೆಲ್ ಅನ್ಬೋದು. Cells within a cell. So you can notice in three-celled pollen grain, nearly 60% of angiosperm plants, they liberate pollen grain in two-cell stage. The remaining 40%, they liberate the pollen grain in three-celled stage. So ಈ pollen grain male gametophyte ಬಗ್ಗೆ discuss ಮಾಡೋಣ. The liberated pollen grains, they have to fall on the stigma. If they don't fall on the stigma, they become non-viable. ಇವು ಅವು ಪ್ರಾಪರ್ಟಿ ಕಳ್ಕೊಂಡ್ಬಿಡ್ತವೆ. ವ್ಯಾಲಿಡಿಟಿ ತುಂಬಾ ಕಡಿಮೆ ಇರುತ್ತೆ ಪೋಲನ್ ಗ್ರೈನ್ಗೆ. ಸೊ ಅದೇ ಸ್ಪೀಶೀಸ್ ಪೋಲನ್ ಗ್ರೈನು ಬೇರೆ ಗಿಡ ಬಟ್ ಸೇಮ್ ಸ್ಪೀಶೀಸ್. ಈಗ ಹೈಬಿಸ್ಕಸ್ ಪೋಲನ್ ಗ್ರೈನು ಸ್ಟಿಗ್ಮಾ ಆಫ್ ದಿ ಹೈಬಿಸ್ಕಸ್ ಫ್ಲವರ್ ಆಫ್ ಅನದರ್ ಪ್ಲಾಂಟ್, it should fall on the stigma, only then they undergo germination. ಪೋಲನ್ ಗ್ರೈನ್ ಜರ್ಮಿನೇಟ್ ಆಯ್ತು ಅಂದ್ರೆ ಪೋಲನ್ ಟ್ಯೂಬ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ಸೀಡ್ಸ್ ಜರ್ಮಿನೇಟ್ ಆದರೆ ರಾಡಿಕಲ್ ಅಂಡ್ ಪ್ಲಿಮ್ಯೂಲ್ ಅಂತೀವಿ. ಪೋಲನ್ ಗ್ರೈನ್ ಜರ್ಮಿನೇಟ್ ಆದರೆ ಪೋಲನ್ ಟ್ಯೂಬ್ ಆಗುತ್ತೆ. ಓಕೆ, ಈ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ನೋಡಿ, ಇಲ್ಲಿ ಡಿಫ್ರೆಂಟ್ ಸ್ಟ್ರಕ್ಚರ್, ಡಿಫ್ರೆಂಟ್ ಶೇಪ್ ದೇ ಆರ್ ಹ್ಯಾವಿಂಗ್. ಓಕೆ, ಸೊ ದಿ ಪೋಲನ್ ಗ್ರೈನ್ ಅಲ್ಲಿ ಕೆಲವಲ್ಲಿ ಸ್ಪೈನಸೆಂಟ್ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ಇರುತ್ತೆ, ಕೆಲವಲ್ಲಿ ರೆಟಿಕ್ಯುಲೇಟ್ ಪೋಲನ್ ಗ್ರೈನ್ ಆರ್ನಮೆಂಟೇಶನ್ ಇರುತ್ತೆ. ಓಕೆ, so these are the different types of ornamentation that you can notice in pollen grain. So pollen grain ಇನ್ನೊಂದು ಹೆಸರು male gametophyte. So the pollen grains have to land on the stigma before they lose viability. ಈ ಎಕ್ಸ್ಪೈರಿ ಪೀರಿಯಡ್ ಇರುತ್ತೆ ನೋಡಿ, ವ್ಯಾಲಿಡಿಟಿ ಪೀರಿಯಡ್. ಇಲ್ಲಿ ವ್ಯಾಲಿಡಿಟಿ ಅಂತ ಬರೆಯಕ್ಕೆ ಹೋಗ್ಬಿಡಿ. ವಯಬಿಲಿಟಿ ಪೀರಿಯಡ್ ಆಫ್ ಪೋಲನ್ ಗ್ರೈನ್ ಇಸ್ ವೇರಿಯಬಲ್. It depends on the temperature and humidity of that particular day. So the viability ಅಥವಾ the fertility of the pollen grain, it depends on temperature and humidity. So the viability of pollen grains of some cereals ಅಂದ್ರೆ ಧಾನ್ಯಗಳು, ಭತ್ತ ಗೋಧಿನಲ್ಲಿ 30 ನಿಮಿಷ ಇರುತ್ತೆ. 30 ನಿಮಿಷ ಆದಮೇಲೆ they cannot produce the pollen tube. Some members of leguminosae, Fabaceae family, Rosaceae, ಮತ್ತೆ ಪೊಟ್ಯಾಟೊ ಫ್ಯಾಮಿಲಿ, ದಟ್ ಇಸ್ ಸೊಲನೇಸಿ, ದೇ ಹ್ಯಾವ್ ವಯಬಿಲಿಟಿ ಫಾರ್ ಮಂತ್ಸ್. ಸೊ ಪೋಲನ್ ಗ್ರೈನ್ ರಿಲೀಸ್ ಆಗಿ ಒಂದು ತಿಂಗಳಾದ್ರೂ ಏನೂ ಆಗಲ್ಲ ಅಂತ ಪೋಲನ್ ಗ್ರೈನ್ ಅಲ್ಲಿ. ಕೆಲವು ಪೋಲನ್ ಗ್ರೈನ್ಸು 30 ನಿಮಿಷ ಅಷ್ಟೇ ಇರುತ್ತೆ. ಈ ಧಾನ್ಯಗಳಲ್ಲಿ ಇರುವಂತ ಪೋಲನ್ ಗ್ರೈನ್ಸು 30 ನಿಮಿಷ ಅಷ್ಟೇ ಅವು ಆಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ. ಅದಾದ್ಮೇಲೆ ಪೋಲನ್ ಟ್ಯೂಬ್ ಫಾರ್ಮ್ ಆಗಲ್ಲ. ಆದ್ರೆ ಲೆಗ್ಯುಮಿನೋಸೆ, ಫೆಬೇಸಿ, ರೋಸೇಸಿ ಮತ್ತೆ ಸೊಲನೇಸಿನಲ್ಲಿ ದೇ ಆರ್ ಆಕ್ಟಿವ್ ಫಾರ್ ಮಂತ್ಸ್ ಟುಗೆದರ್. ತಿಂಗಳಾಂಟ್ಗಟ್ಲೆ ಆಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ. ಅಟ್ ಎನಿ ಪಾಯಿಂಟ್ ಆಫ್ ಟೈಮ್ ಇಫ್ ದೇ ಫಾಲ್ ಆಲ್ಸೋ ದೇ ಡೆವಲಪ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ದಿ ಪೋಲನ್ ಟ್ಯೂಬ್. The economic importance of pollen grains. See, pollen grains, they are rich in nutrients. So a honeybee, they not only collect nectar from the flowers, they also collect pollen grain. ಸೊ ಈ ದೇ ಆರ್ ದಿ ಬ್ರೆಡ್ ಆಫ್ ದಿ ಹನಿ ಬೀಸ್ ಅಂತಾನು ಕರಿತೀವಿ. ಸೊ ದಿ ಪೋಲನ್ ಗ್ರೈನ್ ಆರ್ ರಿಚ್ ಇನ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್. Pollen tablets are used as food supplements. Pollen tablets and syrups increase the performance of athletes and race horses. ಸೊ ಈ ಸ್ಪೋರ್ಟ್ಸ್ ಅಲ್ಲಿ ಪಾರ್ಟಿಸಿಪೇಟ್ ಮಾಡುವಂತ ಪ್ರೊಫೆಷನಲ್ ಪ್ಲೇಯರ್ಸ್ ದೇ ಟೇಕ್ ದಿಸ್ ಪೋಲನ್ ಟ್ಯಾಬ್ಲೆಟ್ಸ್ as they are believed to increase the performance of the sports person, as well as even in race horses they are fed with this pollen tablets as a food supplement. Okay, so along with general food they also take this pollen tablets as a food supplement. So the pollen grains, they can be stored for years together, for eternity ಅನ್ಬೋದು. You can store them in liquid nitrogen. See, nitrogen is in gaseous form. For them to be in the liquid form, the temperature should be minus 196 degrees centigrade. The pollen grains, na, we can store them for eternity in liquid nitrogen. So the pollen grains are used as pollen banks in crop breeding programs. So pollen grains, they are all used as pollen banks. ಒಳ್ಳೆ ಗಿಡದ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ನ ಇಟ್ಕೊಂಡು ಕ್ರಾಪ್ ಬ್ರೀಡಿಂಗ್ ಪ್ರೋಗ್ರಾಮ್ಸ್ಗೆ ಈ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ನ ಪೋಲನ್ ಬ್ಯಾಂಕ್ಸ್ ಆಗಿ ಯೂಸ್ ಮಾಡ್ತೀವಿ. So pollen grains of some plants like Parthenium or carrot grass. So Parthenium ಅಥವಾ ಕ್ಯಾರೆಟ್ ಗ್ರಾಸ್ ಅಲ್ಲಿ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ಇರತ್ತೆ ನೋಡಿ, ಕಾಂಗ್ರೆಸ್ ಗಿಡ ಅಂತೀವಿ ಇದನ್ನ, ಪಾರ್ಥೇನಿಯಂನ. ಸೊ ಈ ಪಾರ್ಥೇನಿಯಂ ಅಥವಾ ಕ್ಯಾರೆಟ್ ಗ್ರಾಸ್, ಆ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ಅಲರ್ಜಿಕ್ ಫಾರ್ ಸಮ್ ಪೀಪಲ್. ಸೊ ಆ ಫ್ಲವರಿಂಗ್ ಸೀಸನ್ ಬಂದು ಜುಲೈ ಟು ಅಕ್ಟೋಬರ್ ತನಕ ಇರತ್ತೆ ಈ ಪಾರ್ಥೇನಿಯಂ ಗಿಡದಲ್ಲಿ. ಸೊ ಆ ಸೀಸನ್ ಅಲ್ಲಿ ಕೆಲವರಿಗೆ ಆಸ್ಮಾ ಸಿಂಪ್ಟಮ್ಸ್, ಬ್ರಾಂಕೈಟಿಸ್ ಇವೆಲ್ಲ ಇರುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ಬಿಕಾಸ್ ಆಫ್ ದಿಸ್ allergic to this pollen grains of Parthenium. So this leads to chronic respiratory disorders like asthma, bronchitis, etc. ಸೊ ಇಷ್ಟನ್ನ ಇವತ್ತು ತಿಳ್ಕೊಳ್ಳೋಣ. ಈ ಕಾನ್ಸೆಪ್ಟ್ಸು ಇಷ್ಟನ್ನ ಸಾಕಾಗತ್ತೆ ಇವತ್ತಿನಿಗೆ. ನಿಮಗೆ ಇದರಲ್ಲಿ ಏನಾದ್ರು ಡೌಟ್ಸ್ ಇದೆಯಾ? ಸೊ ಡು ಯು ಹ್ಯಾವ್ ಎನಿ ಡೌಟ್ಸ್ ವಿತ್ ವಾಟ್ ವಿ ಹ್ಯಾವ್ ಡನ್ ಸ್ಟಿಲ್ ನಾವ್? ಏನಾದ್ರು ಡೌಟ್ಸ್ ಇದೆಯಾ ನಿಮಗೆ? ಡೌಟ್ಸ್ ಇದ್ರೆ ಮೈಕ್ ಆನ್ ಮಾಡ್ಕೊಳ್ಳಿ, ಕೇಳಿ.